No? No?
Hello?
Can you hear us?
Yep. Now I can
Okay,
hear.
cool. Cool.
That would have been unuseful.
Yeah. Cool.
Okay.
Okay.
So any
Uh,
announcement?
yeah, so I was gonna say I should have posted all of your homework grades on Gradescope, so please check that and make sure everything seems good to you. So, and if there's like any concerns, reach out to me. Yeah, that's it.
Okay. So Ekin, you know what's funny today?
What?
I didn't have to prepare for class. You know why?
Why?
I think somebody else is gonna talk today.
Guest lectures.
Yeah.
Uh,
So do we have a list? Who's on first?
yeah.
And
So
who's
the
gonna keep the
first,
time?
I can keep the time, and each of you will get like 20 minutes, or 15 minutes to present, 5 minutes for questions. So we'll try to keep track of all of them. And the first group is Maxwell, Katie, and Daniel. So if you guys want to, let me check that.
How long do they have? 15?
50,
Just five?
15. Yeah,
Okay.
1-5. Yeah, you
So
guys should.
people get both evaluation for what they present as well as evaluation for the question they ask. So if you have good questions, we're going to note that. That's for those that are listening.
Cool. Yeah, I guess you guys can get started. Yeah.
Okay, go
Yeah.
ahead.
Go ahead.
Sounds good. Thank you. All right, so yeah, we're gonna be presenting on motion correction techniques in MRI. To get started, we'll talk about the problem. There's a lot of causes of motion. You know, babies could be moving in a scan, people could be breathing, hearts could be beating, and that will cause motion artifacts. Some example of some that we might have already seen is like rigid motion, like when you're shaking your head side to side would cause some blurring artifacts. As well as if you're imaging during breathing, breathing could cause uh, like aliasing and blurring as well. So these are things that we want to try to avoid. It kind of breaks up into two main types of motion compensation. The first one is prospective, which involves kind of using your knowledge of the current motion state in order to improve your acquisition during a scan. And then retrospective is more of a hindsight technique, where you acquire your data and then you retrospectively um, fix it given the motion state, right? So we're going to also break down some common approaches that people have taken. So sensor-based is kind of trying to sense what the motion state is like with something like an ECG signal, a breathing monitor, pilot tone, which we'll get into. And then there's these data consistency methods where it's more acquiring your case space and then uh, setting up a model or a reconstruction in order to get a nice image. So let's start with the sensor-based. Gating is a really common technique. Specifically, this is respiratory gating, where the blue signal shows the breathing signal, and we acquire data in an expiration, or when there's not a lot of motion due to breathing going on, right? And that's one way to decrease motion artifacts. It's not just limited to breathing, but also for cardiac motion. We could do the same thing, but timing between heart pulses, right? So the heart's not moving too much. Navigators are another technique that are used where you have a small pre-scan over some small regions that are defined by the yellow rectangles. And these regions will give you information of heart motion or lung motion in the top and bottom graphs respectively, right? So these are kind of just some common approaches that have been used so far, as well as breath holding, right? This one's pretty simple. You just have your patient hold their breath while you take an image. Some downsides here are that there's a lot of synchronization required and not everybody could hold their breath for a long time. So I'm going to be diving more deeply into the pilot tone method, specifically for free breathing radial imaging. We'll start off with what is a pilot tone. Simply put, while you're taking an MRI image, you turn on a constant frequency and
MR image.
MR image. Thank you. While you're acquiring an MR image, you turn on this constant tone that interacts with the human body And then our signal equation is going to look something like this. It's the original signal that we would get without the pilot tone plus this pilot tone, right? In a Cartesian acquisition, this is going to lead to a zipper artifact, which doesn't look very advantageous until we tell you that this pilot tone strength is actually modulated by the breathing signal, right? So when we acquire this pilot tone, it's going to give us an indication of the breathing signal. More specifically, we can take our k-space and take a Fourier transform along the readout direction. We get a constant frequency pilot tone, right? And its amplitude will actually be modulated by the respiratory signal. And that's how we could extract the respiratory signal using a pilot tone. So in recap, kind of a step-by-step, is we want to turn on this constant off-resonance frequency. We acquire the MR data with a slightly increased field of view to make sure that the pilot tone doesn't interfere with the actual image. Then we can extract this motion signal from the MR acquisition and then finally reconstruct the image retrospectively or use it for gating prospectively. We're going to focus on the former. Retrospective reconstruction is what was covered in this paper. More specifically, they took this pilot tone signal that gives us in turn a respiratory signal and binned it into different motion states. Given that this respiratory signal is periodic, they bin it into like low motion states to high motion states, which represents bins 1 through 10. And we're not going to get into too much detail here, but they view this motion, these bins rather, as an extra dimension. The motion dimension or motion states is another dimension in our acquisition. They throw it into an optimization problem, and then they're able to retrospectively correct the motion images. And here's some examples of that. So on the left is just typical like motion averaging, and on the right is using this extra dimensional reconstruction. And the yellow arrows kind of point your attention to where there are areas of higher detail. So here's another image. Yeah, we could definitely see much more detail and sharpness compared to motion averaging. So that's, you know, a sensor-based method. Here's Katie's going to dive into a data consistency method.
Okay, so I'm going to be going over Tamer, which stands for Targeted Motion Estimation and Reduction. It's a retrospective motion correction method that provides a joint estimation of the desired motion-free image and the associated motion trajectory. So Tamer attains these estimations by minimizing the consistency error of a modified sense-forward model. This method was developed by Melissa Haskell, Stephen Colley, and Lawrence Wall. I will be going over the algorithm as well as the results as discussed in their paper. Next slide. Okay, so how it works. So the Tamer method uses a modified sense model. So it looks a lot like the one we've seen in class, but there's some differences. So the main differences that you can see in this picture is that for each shot of the multi-shot sequence, right, the submatrix basic is the encoding matrix, right? And for a particular shot, and it's dependent on a couple of things that might be new. So TXY, right, in that picture, you'll see that's the translation, the in-plane translational operator. TZ is the through-plane translational operator, and then lastly, R is the rotation operator. And this is all so that we can track the motion. So this is how we have to, what we have to add to our model in order to actually, you know, optimize for both the motion trajectory and for the image itself. All right, next slide. Okay, so in order to make the joint optimization more manageable, and as you probably noticed in the last slide, trying to solve that is going to be a pain and probably not, you know, efficient. Really not efficient, actually. And so, in order to make it more manageable, the Tamer method actually came up with a new way of going about optimizing. So here we have an encoding matrix, right, the E, and they're basically split into two. So these two submatrices, right? And so for the encoding matrix that is associated with the vector xt, which is the target voxel values. And then there's also the encoding matrix associated with xf, which is the fixed voxel values. And I'll be going into a little more detail what I mean by that in the next slide. Oh, not yet, Daniel. But the target voxels are basically selected every iteration. But for the fixed voxel values, those ones are fixed based off of the initial, basically initial scan. And so that I'll go over that in the next slide. Now next slide. Okay, so essentially what we have here is the first part is the initial reconstruction. this is the part where we basically get our fixed voxel values. And what I really mean by that is basically they're going to choose a bunch of target voxels, right? Target voxel values. And these are the ones that we're actually going to change through each iteration. Whereas the fixed voxel values, they're going to hold, they're just stay the same. They will update occasionally, but for the most part, stay the same. So this is actually what makes the model a lot faster. We only have to deal with something changing, a small amount changing, about 5% of the total voxels. So the reduced model is most effective when voxels that are strongly coupled to one another are also optimized together. So essentially, we want to take the target voxels that have the most, that are basically the most they're interacting the most with one another. We want to take those and make sure that those are the target voxels that we're selecting. So we want to pair them up together. And so the Tamer algorithm is therefore performed using only the coil groups with the largest correlation artifacts. So you see that in this picture, right? We have the sensitivity maps of the two coils here. And essentially what we have is the error, and we get the correlation map, the correlation model of between them, right? And you can see that we want that white line in the middle, or the diagonal, right? That's basically saying it's correlated with itself. But all the parts, all the white spots that are outside of that, that's the correlation with, not just itself, with other coils. And we essentially want to perform the Tamer algorithm over the coils with the most, that are most interacting. All right, next slide. Okay, so essentially the way we pick our first target voxel, right, is by basically, they usually take the center, and from there, they take the correlation, matrix, and they basically calculate which voxels would be the most interacting with this one. And from there, those are the ones that they choose, and they only choose so many of them. So now that we have the initial target voxel, we attain the initial guess of the patient's motion, which is determined by evaluating the data consistency metric over a range of values for each of the motion parameters. The best value for each parameter is selected to construct the initial guess. And then the basic algorithm is as follows. So they iterate through this until we either hit the max number of iterations or we're within a certain error. We estimate the signal generated by the target voxels using the reduced model described previously and update the signal. Then we estimate the vector of target voxel values also using the reduced model. Then we solve for the error of the current iteration, and we update the target voxels by shifting them perpendicular to the phasing code direction, and then we get a new estimation based off of the gradient, and then we start over. And we keep doing that until we basically reconstruct our desired image, all right, or in desired estimate. Next slide.
Hold on, is the model rotation translation, affine? What is it for motion?
I can I show you it during the question part, because we're kind of
sure.
Sorry, yeah, I have that if at the end. Okay, so disadvantages and advantages and disadvantages, right? So this is a retrospective method. Essentially, we don't have to change the scan procedure. We just can scan the normal way. Tamer significantly reduces computation time for past algorithms that have tried to solve this. The downside is the current implementations are still kind of lengthy. They are essentially, and also Tamer requires like multi-channel data. So you kind of, we need basically additional degrees of freedom, which is what is provided by multi-channel acquisition. And then lastly, the Tamer algorithm assumes static coil profiles. So for larger motions, we can't really do that. All right, next slide.
Okay, so now we're going to talk about the last method that we wanted to cover, which is called Namer. But before we start talking about Namer, we need to talk a little bit about what a neural network is, because that's what this approach is based on. So in the last 10 years or so, we've seen a deep learning boom where neural networks and machine learning approaches have shown to be super effective in solving many different problems in many different fields. But some ways in which this is applied to MRI, I'll just list two of them in particular. One of them is kind of doing this compressed sensing approach that maps between undersampled data and fully sampled data. Another way you can use a neural net is by learning the parameters to some sort of model in order to help with reconstruction. And this is particularly what we're going to focus on. Next slide. So Namer stands for Network Acceleration, Estimation, and Reduction. This is a sequel to the Tamer paper that introduces neural nets to significantly speed up calculation and also improve accuracy. Specifically, we have a convolutional neural network, henceforth I'll call it a CNN, which learns the artifacts in an image. It then removes these artifacts by subtracting them from the original motion corrupted image, tries to now estimate the parameters for the sense-based model that Katie was talking about earlier, and then reproduce an image in order to continually, iteratively remove all the artifacts. Next slide. For a little more detail into each part, each image that the model is trained on is normalized and broken into patches. Each patch is fed into a CNN, which is a specific neural net that is really good at analyzing and understanding visual information. As you can see in this picture below, on the left, we shove in an MR image. And on the right, you can see that it spits out the artifacts that it learns. And in the top right, we do the simple subtraction in which we remove the artifacts from the input to get this estimate of a motion-free image. Next slide. So Namer is also using the sense-based motion-forward model in order to incorporate the idea of motion in our reconstruction. And as we saw, there is this issue where this joint optimization is very complicated to do. But what happens is that because we have a CNN incorporated into the joint optimization, it not only improves the accuracy of the reconstruction, but also gives us right off the bat the estimation for our target voxels or our images. This means we can kind of sidestep part of the optimization by ignoring the fact that we also have to find out what the voxels are. This means that we can just directly optimize the model parameters, and beyond that, actually separate them out for each shot or each line, depending on how the acquisition was performed. So this means you can massively parallelize the computation, bringing it from about 50 minutes in Tamer to like 7 minutes in this approach. Next slide.
Hey, can you wrap up in like 2 minutes?
Yes.
Possible.
Yes.
Okay.
Okay. Okay. So then we take these new parameters and we perform a simple least squares estimate to find the image that best matches what the model would predict and how that matches with the case-based data. We get this resulting image that is supposed to have no more artifacts. We feed it back into the CNN and we keep looping until we get something that doesn't have artifacts. Next slide. It turns out that so they trained and tested this on 2 types of data, both synthetically motion corrupted data and also real in vivo data. And in both cases, it was able to significantly remove artifacts and also decrease error in the reconstruction. It can't fully remove everything. There's still some bringing and blurring, but as you can see in the left-hand column of the bottom image, for something that has severe corruption at about 43% error, in the second column, we got it down to about 11 or 12% error. And it also converges way faster than other methods. Next slide. So to kind of recap a little bit, we talked about 3 methods: pilot, tone, tamer, and namer. Pilot tone was a sensor-based approach that uses an external RF tone to extract the signal. It's great because it doesn't require contact with the patient and can be used both retrospectively and prospectively, but it does need extra hardware. The thing about Tamer and Namer is that they are both retrospective data consistency-based approaches. Katie already talked about Tamer quite a bit. I'll talk a little bit about Namer. It has many of the same pros and cons, but some extra pros are that it is even more effective and much faster by an order of magnitude, while some limitations are that it does require GPUs, and there's a question about generalizability. Final slide. Okay, so just a few considerations about what method should you actually use. There's obviously cost and practicality. Each method has its own implementation challenges. If you can tell someone to hold their breath and that works for moving respiratory artifacts, then that's fine. That's all you need to do. But for some of these other methods we talked about, like with data consistency, they're not yet widely clinically implemented, if implemented at all. You won't see a neural net when you go in for a scan. It's mainly relegated to research. And finally, the types of artifacts that you might expect, you need to think about like, what are you imaging and what can you hope to mitigate? If you, for example, are just expecting to deal with a respiratory artifact, maybe you can get away with a navigator or a breathing monitor. But for more complex types of motion, such as rigid body motion, you would need something more complex. And last slide, which method is best? As we've seen in this class, it depends. Finally, just some acknowledgements. Big thanks to Mickey and Ekin for running the course and all the time and care they put in. Thank you to Suma, one of the grad students, for providing us with Peloton data and connecting us with the author, Melissa Haskell. And thank you everyone for listening.
Okay, questions?
Oh, sorry.
I actually had one question. I was curious, like one artifact we've seen a lot in the class for motion is like when eyes move and you can kind of see the replications. And I know like eye tracking is a really robust technology that's used in like VR and AR. I was wondering if you guys saw any examples of like the use of eye tracking for like motion correction, like prospectively, or anything like that.
Funny enough, no, not really. Actually, I haven't seen, at least for these methods, not so much. I would think that that would probably, I don't know if like a Tamer algorithm would work. I mean, that's an interesting question. I don't think I've seen a lot of various implementations of that, but I would think that would that be considered rigid motion? Maybe this would work for something like that. But I guess something to see in the future.
At least in the papers I saw, they mainly focused on the more common types of motion artifacts like cardiac and respiratory. So there wasn't as much information on eye tracking or eye movement, but that is definitely, you know, a very interesting approach we could look at.
Would you like to see the slide with more, Nikki, that has the full? I had it muted because it was a little too much. But if Daniel, if you want to go back to the slide, it's like slide 36 with the actual equations on it. So you could, I know I had to kind of rush. Sorry, it was a little. Our presentation was long, if you have any questions, I can do my best to answer.
What's the motion that it tries to correct for? I mean, is it global motion? Is it local? What is it?
Generally, well, for this paper, rigid motion, head rotation, right? Yeah, that's considered rotation.
Rotation translation?
Yeah.
Okay.
Yeah.
So does it answer then the question about the eye movement? Is that really would have been global rotation translation, or would be more local rotation translation?
I think it would be more local, but I don't necessarily know that this wouldn't work for that though. So that's where we'd have to look into it. I don't think they have looked into it.
Any more questions?
Well, I have a question. So when you guys were looking into like different methods, what did you see kind of like the differences in methods depending on like what body, like I guess part of the body they were imaging, or what part of the body's motion they were trying to correct overall? Like, did you see some differences there?
Yeah, from the looks of it, it seems like the repetitive motion types, like breathing and cardiac gating, seem to be like the most common. And then for rigid body motion, kind of what Katie and Max were covering, more so like these more model-based reconstruction techniques seemed a lot more common. If that answers the question.
Yeah.
Yeah.
Yeah, more questions.
Yeah, I had I imagine this applies more to neural nets, but I'm also not sure. But as far as when you are like correcting artifacts with this model, I understand you had like a ground truth to go off of, but like obviously you don't always have a ground truth. So did they talk about how you would quantify the error that your artifact correction introduces versus the error it reduces? Like how do those play off each other, if that makes sense?
Right. So the idea of kind of the neural net is there has to be paired data in the sense that you have an input matching to an output. Here, because we're matching between an image that has motion and doesn't have motion, it's a little harder to think about how you can acquire that data, because if you acquire it once without motion, once with motion, those aren't really the same anymore. So what they actually did is they took motion-free data, and at least for how they trained the model, they trained it on synthetic data in which they augmented the motion-free data by applying the sense-forward model to introduce motion trajectories. This means that you had data that had motion, but you already knew what the golden standard like ground truth was, because the motion was generated from the ground truth. So then the network will try to figure out what the artifacts are, and then take the motion corrupted image, subtract out the ground truth, and look at that, which is the actual remaining artifact, and compare that with what the network pulls out. And then that's how it would train it. There is some question about, is it proper to use synthetic data versus like real data? But in the results, it was seen that even if they train it on in vivo data, which actually has motion corruption, where they instruct the person specifically to shake their head, it was still able to reduce motion artifacts and error significantly. So it seems like it still is able to account for that.
Okay. Thank you very much. I think that's it, right? That's the 20 minutes. Perfect.
Yeah.
All right. Well, thank you all for presenting. And hold on. Oh. All right. So where was I? Yeah. Who's the next group?
Yeah, it's Tiffany, Amit, and Mark. Next group.
Please go ahead.
I'll let you guys know when you have 2 minutes again, FYI.
Sounds good. Thank you.
Okay.
All right. Hi, everyone. So we're going to be presenting about magnetic resonance fingerprinting today, which is a method for quantitative MRI. And so most MR images you see are qualitative, like, for example, this T1 weighted image over here. And the contrast here kind of tells us the relative difference in T1 values in the image. But if you look at just a single pixel on its own, then sort of the intensity value is kind of meaningless. It does depend on T1, but it also depends on the pulse sequence you used and your scanner. And the question is, what if we actually want to know the true T1 value at each pixel? This is the goal of a quantitative MRI. So the goal is to basically, at each pixel in your image, get a set of quantitative MR parameters. So these parameters can be things like T1, T2, proton density, etc. Now, why you might want to do this? Well, one is discovery, right? If you have some new material and you want to know its underlying MR parameters, you could use a quantitative MRI method. The second thing could be its use as a disease indicator. So on the right here is a study that shows that T1 values in the brain can be correlated with disease states. And finally is robustness. So there's certain situations where qualitative MRI may fail. So think about a situation where I globally increase all of the T1 values of some image. Because a qualitative MRI is relative, it might miss something like that. So let's start by looking at sort of a traditional quantitative MRI method. So here is T1 mapping. Basically what you do here is you use an inversion recovery sequence. So you hit your magnetization with a 180 to flip it. And as it recovers with T1, you sort of sample images at each sort of point here shown on this curve, right? And so now to recover your T1 at a particular pixel, you just track a pixel through the sequence of images that you get. And then you fit that to an exponential, and that gives you your T1 parameter. And so in some sense, what happens here is that you get a set of T1 weighted images, which sort of encodes T1 information. But the problem here is now if I want T2 or some other MR parameter, I'm kind of stuck because I only have information about T1, right? I have to do a whole nother sequence, have to get another whole set, another sequence of images in order to get other information. So ideally what we want to do is encode multiple types of information in a sequence of images, right? So here, this is the sort of idea of MRF. We're going to take some mystery pulse sequence, which we'll talk about later. But suppose this pulse sequence gives us a sequence of randomly weighted images, shown here on the bottom. Intuitively, that's going to encode information about all of our MR parameters, right? But now look, if we track sort of one pixel through this sequence of randomly weighted images, we're going to get something that looks random, right? But I would argue that this actually is a function of all of the MR parameters at that pixel, right? Sort of has mixed up all this information. And so now if I were to move this pixel to a different location and to a different material with different underlying MR parameters, this sort of squiggle changes, right? And so we're going to call this squiggle, right, that you see this little 1D signal, the fingerprint at that pixel. And sort of it's called the fingerprint because it's an identifier of the material or the underlying parameters at that pixel. And so now the question is, how do I disentangle this squiggle into the parameters I want? Well, one idea is, as I mentioned through the name fingerprinting, is to build up a database of known fingerprints and their sort of corresponding MR parameters. So here I show for a bunch of common materials what their fingerprints might look like. Now, if you have a database like this, you can get some new material, look at the fingerprint and see how well it matches up to one of these existing materials. And that'll tell you what the parameters are. So now this is sort of like the overall approach of MRF. First, you build up a dictionary of known fingerprints and their corresponding MR parameters. Then you have some new object. You get the fingerprints at each pixel using this sort of mystery sequence. And then you match and you say, hey, which fingerprint my database best matches this? And then you use the MR parameters corresponding to that. And so you can get them all simultaneously with one sequence. Now, so I left some questions unanswered, and these are sort of what I'm going to talk about are the main research directions within MRF. So the first is I didn't talk about that sequence. So how would you actually design a sequence that allows you to encode all this information and get unique fingerprints for each parameter set? The second is how do you actually build up this dictionary or database of fingerprints? And then how would you actually match a new one to the entries in that? And then finally, is this actually useful? I talked a little bit about a potential clinical application earlier, but can you actually use MRF in practice? And so Tiffany is going to go ahead and answer the first two questions.
All right, so the first question is about the MR sequence design. And so we are mixing up all the parameters in this funny squiggle, but we need to mix them up in a way that can be unmixed. And the key idea to making that possible is that every material's fingerprint has to be unique. So what kind of MR sequence will give you a unique fingerprint for different materials? In MRF, we use random acquisitions. So what I mean by this is that we start with some base pulse sequence. In the original paper, they used balanced SSFP, but we randomize all the different parameters of the sequence, such as the flip angle, TR, TE, etc. So if we look at this picture, there's a series of acquisitions, a series of excitations. Each one, in each one, a readout is taken, but they all have a different flip angle and TR. And these are chosen in a pseudo-random sequence. And so the idea intuitively is that randomness will naturally produce independent and uncorrelated measurements, which will make the fingerprints unique. Another way to think about randomness, and also an advantage of randomness, is that it is tolerant to artifacts such as undersampling and motion. So if we think about the artifacts that we've seen in this class, we know that artifacts are a result of the sequence of the different sequence parameters. But I just said that the sequences are random relative to each other, which means that their artifacts have to be uncorrelated relative to each other. And so the only underlying signal remaining between the different sequences is the actual signal that we wanted to measure. So more specifically, if we look at a single acquisition from MRF, it's extremely undersampled. So this is an undersampled spiral acquisition, and we see these very structured undersampling artifacts. But if we do the entire MRF acquisition and do a bunch of different sequences, so in this case, there were 1,000 different sequences taken with different TR. The blue line represents the signal that we measure, and the red line represents the fingerprint that we were trying to match it to. So we want them to look about the same shape, which they are, but we see that the blue signal is much noisier. However, it still matches the shape, and all of these undersampling artifacts just look like noise. So because the sequence is random, these kinds of artifacts don't interfere with the fingerprint matching. Okay, so now that we've talked about how to do the MR design, we can talk about how to build the dictionary. So this is pretty straightforward. You basically choose a bunch of values of parameters that you want to test. In this case, they were doing brain imaging, and so these were some of the possible T1 and T2 values, and their dictionary had about 500,000 entries. And so you take each of these entries, you plug them into the block simulator, and you get fingerprints out. So each set of parameters goes through all of these 1,000 different acquisitions, and you measure how much signal comes out. And that corresponds to the fingerprint of that set of parameters. So we have the dictionary, now we try to match to it. So in the original paper, they use a really straightforward approach. For each of the voxels in your image, you do a comparison with every single dictionary entry, and you compute the inner product, and you choose the one that was the closest. So this is not a particularly efficient way to do it, and you can only have discrete values to match to in the dictionary. So research has gone into better ways to perform this matching. One idea is to compress the dictionary using something like an SVD, and then it can be faster to match because you're working in fewer dimensions. Even fancier than that, instead of using a simulation and a dictionary, we can try to directly invert the block equations. So you can write MRF in a framework known as compressed sensing, and then use optimization, basically use linear algebra, to try to invert the block equations. Or you can try to use a neural network that is trained in simulation, so basically using your dictionary, and it will also learn how to invert the block equations. So now we've covered basically all of the details of how MRF works. So here is a picture of overview. So first we do the acquisition, and we said that we used a random set of acquisitions. Then we generate the dictionary in simulation, and then we match those two things together using some kind of algorithm. And after the matching, we are able to get these quantitative maps. So I'm going to pass it over to Mark now, who's going to talk about different clinical applications.
All right, so the third major question of MRF was, how is it useful in the real world? And there are sort of three major paths forward for MRF. The first is to complement current MR scanning techniques. As you can add a quantitative aspect to your scan, or you can replace current scans in certain situations. The second way forward is if you want to screen for different diseases. If you have different MR markers that indicate or so uniquely identify different diseases, you can scan over a volume and see if those pop up, and then flag that as a potential thing to follow up with. And the third major area is tissue classification. So if you have a disease state or a healthy tissue that is uniquely identified by a certain set of MR parameters, you can start to automatically segment, so like brain scans for tumor and non-tumor with just a computer based on the quantitative results of your scan. Next slide. All right, so then the question is, what has the research really focused on? And most of the work has been on showing that MRF is a feasible technique compared to current MR designs. So in these images, you can see a MR, a CT, and MRF scan, and all of them show two tumors in an individual's liver. And in addition to just imaging, you need to show that the T1, T2, other MR values that you get out of the scan are corresponding to the previously published work. So in the liver, you expect a certain T1, T2 values to exist. And do you get that with your MR scan? Is it repeatable? And the body of work so far has shown that it is, and it is repeatable, and it overwhelmingly agrees with previously published work, but that still needs to be more and more established in large areas of the body. Success has been shown in brain scans, abdominal scans, both with breath holds and free breathing, and heart scans with heartbeat synchronization. So in a lot of cases where traditional MR can be used, MRF can also be used. Next slide. As far as clinical work that goes beyond traditional MR scanning, this is an example study where they were able to detect and classify different regions in a person's brain with epilepsy. So on the right, you can see that the MRF scan was able to detect an epileptic lesion. It's very faint that the traditional MR scan was not able to find. So sometimes you can find things that are harder to find with a MRF scan than you can with an MRF scan. In addition, on the left, you can see how MRF, the quantitative extraction allows you to differentiate materials. So you can see the pink clump is white matter, and then the red clump is an epileptic nodule. You can also differentiate between non-epileptic nodules and epileptic nodules, so like diseased and non-diseased areas. And this is a statistical correlation, as there's lots of variance, but as you narrow in on the sort of parameters and statistical spread of these various areas of the brain, you can start to classify these areas accurately. Next slide. All right, so like any MR technique, MRF has its own clinical challenges. By far, the biggest one right now is the reliability needs to be established. You need to build up this large body of work that shows that MRF is as capable and as reliable as current MR techniques in the clinical setting before you use it on people. The second area is, or second problem is that if you are using it to diagnose different diseases or classify different areas, you need to know what sort of parameters and what the statistical spread of those parameters is across a large and diverse body of people. So you need to go out, you need to actually get that data and acquire that data, know that data before you start diagnosing people with various things. An example of that can be seen on the right, which is from a study of abdominal scans, MRI abdominal scans of individuals, where they went in on the images and they circled an area and said, like, this is the liver, this is the pancreas. And you could see within those areas, there's a large variance in T1 and T2 values. And so you can see how it can be difficult to say, like, oh, this T1 value exactly correlates with the liver, as there's a variance. And so you see, like, okay, the average is different, which between, say, the muscle and the liver they are, but maybe not between the different areas of the pancreas. So you're trying to see which values can be used as indicators of different areas. The third major problem is deformation can cause issues, like with normal MR. Again, MRF can be less sensitive to that than traditional MR, but it can be a problem. And then the fourth issue is that the current generation of radiologists and professionals are not trained to design MRF sequences or use MRF tools or analyze MRF images. And so you either need to go out and train them in using it, or you need tools to convert back and forth between the new and the old to allow them to use it. So the big takeaway is that MRF is incredibly exciting theoretically and clinically. So it the ability to extract exact quantitative information out of an MRI machine is in itself very exciting, but also the clinical applications are very promising and are just starting to be proven and shown out. Companies have started to build in MRF tools. Siemens has an MRF toolkit that you can use for their machines. And the other exciting part is that you don't need to adjust, like you don't need to add hardware or make a new machine to use MRF. You can use it with current generation or possibly future generation, but cheaper MRI machines. With that, I'm going to pass it off to Amit, who's going to do a quick coding example.
Yeah, so we ran a quick simulation.
Sorry, just like if you can wrap up in like 2 minutes, that
this
would
will
be
take
great.
30 seconds. Yeah.
Cool.
So we ran a quick MATLAB simulation of MRF with big thanks to Ekin, who helped a ton with this. Basically, the idea is we did exactly as we mentioned. We first started off by randomizing a sequence. So we started the really simple sequence, just, an inversion pulse and then some random flip angle followed by a spiral readout. So first we randomized the parameters like the TR and the flip angle. Then we made a dictionary. So we basically just had, a double for loop over, T1 and T2 values and filled it out. And then third, we actually acquired this image. This was the same image we saw in one of the homeworks. We used a spiral sequence here to do it after basically every little random acquisition. And then finally, we used the template matching to sort of reconstruct a map of the T1 value. So here I took the bottom row and I made them a different T1 and T2 than the top row, the letters in the top row. And you can see it kind of works, right? It's a little noisy and messy, probably because I didn't do everything 100% right, but it sort of gets the general idea that there's two different T1 and T2s at play. And you can kind of see it actually in the acquisition images. You can see that the bottom letters, at least in the 3 examples I show, are dimmer than the top ones. So those are probably where the T1 weighting was more significant than the T2 weighting or vice versa. But yeah, that's basically it. And here's all of the papers that helped us do this. So shout out to those people too.
Okay, questions?
Yeah, I was wondering if you could comment on like the scan times to perform these. I would assume they would probably be longer than just like a standard anatomical sequence, but maybe I'm wrong.
So that's actually one of the other really nice benefits of MRF is that you can get the same, about similar results to normal MR in the same amount of time, and in some cases even a bit faster. If, for example, in conditions where you have very long relaxation times, where your scan time would start to balloon out as you need to be able to account for that, MRF has been shown to be able to pick that up without having to extend the length of the scan time. So there are actually advantages to using MRF and getting all of these things at once versus doing repeated scans or longer scans.
Yeah, a lot
Yeah.
of the reason that normal scans can take a long time is because they're trying to isolate a single parameter. So you have to like wait for something to decay or wait for something to recover. But because the MRF is mixing it all up, it can actually be fast.
Yeah, and to add to that, you can also get more signal because again, you're not waiting for certain things to happen. You're taking as soon as you flip, you can start your spiral sequence, right? And so that also is a big help.
Cool. That's really interesting.
I guess I have a question. So how would like inhomogeneity, like UN inhomogeneity, affect like your dictionary here? Like, is it going to be a huge problem, or is it okay?
Yeah, it can be. So the cool thing about MRF is that if you account for B1 inhomogeneity, so if you include it as a parameter that you're also estimating, then you can actually use it to help you, right? You can use it as a thing to make your matching better. But if you don't account for it, then yeah, it can skew fingerprints. And the worst possible thing that could happen is it could skew a fingerprint so bad that it matches with the wrong entry. And so you make a misprediction, right? But if you include inhomogeneity as a parameter that you also have in your dictionary, then you can actually predict it as well, along with the other parameters.
And how for actually including it in your you said your dictionary, how exactly is that feasible to actually like accurately come up with that model?
Yeah.
Is that possible?
Yeah, so like as we saw, like B1 inhomogeneity causes off resonance, right, where you don't expect it. And so right, we've seen
B0.
Huh?
B0 inhomogeneity causes off
Oh,
resonance.
B0,
B1
yeah.
homogeneity causes errors in the flip angle.
Got it. Yeah. So you can account for B0 inhomogeneity through off resonance, like we saw in the block simulator. As for B1, I'm not sure exactly how you would do it with like sort of the code we've seen in class, but I would imagine, like Mickey said, it changes the flip angle, so you could sort of account for that by having another parameter, which is at each pixel you have some B1, off like error, I guess, and that increases or decreases your flip angle from what it should be at that pixel. So that's a way that you could account for it and include it as a parameter.
Thanks.
But
Did
Mickey,
you find
what?
any?
No, go ahead.
Yeah. Did you find any papers or works that showed that like using MRF at higher fields, so like 7T versus 3T, like had any effect on this, or it's not like too big of an effect?
I don't know if the other people saw any papers, but I didn't see any papers about this, so I'm not sure.
Actually, there are a lot of papers on 7T MRF.
Does it work better?
Yeah, I haven't checked, but there are some papers.
So I know that it changes the sort of reliability aspect, at least with stuff I've seen where they did different field strength. So you can have, it can change your the distribution you get that you when you're acquiring stuff. So like your it can become like, it can spread out your statistical distribution or narrow it depending on the field strength of different parameters. So it can change your reliability a little bit. I don't think that it's a hard and fast, like increasing your field like makes it worse or makes it better. I think it depends on the specific thing you're trying to do, but yeah.
Thanks.
Ekin, how many groups do we have and how long?
2 more, so we can slowly move on.
Okay, well, thank you very much, the group.
Yeah, thank you for presenting. Cool. Okay, so let's move on to the next group. Cindy. Oh, perfect. Okay. Cool.
Hey everyone. So yeah, today Cindy and I will be focusing on B1 field inhomogeneity and mapping techniques, and kind of going over a few techniques that we found through literature review. So before we get into these techniques, I'd like to introduce the purpose and kind of the theory behind the big B1 field really quickly. As we know, our body is full of protons, and the B0 field aligns these protons to a net magnetization. And then after the RF pulse is what is applied in the perpendicular direction is what flips the spins to the desired angle. And this flip angle scales with the B1 field amplitude as shown in the next equation. And this flipping angle is crucial because the measured MR signals depends on the flip angle of the protons. But again, like I said, this is a theory, and this theory assumes that this B1 field is homogeneous, and therefore all spins in a slice are flipped an equal amount. In reality, though, different areas of a slice see different RF fields, leading to different flip angles. And one reason this occurs is because the RF wavelength is inversely proportional to the B0 field. So RF wavelength decreases when B0 increases. And as shown in the figure on the left, at B0 fields of 1.5 T, RF wavelengths are long compared to the size of the body. But as this main magnetic field is increased, these wavelengths become the same size or even smaller than the region of the body that's being imaged, resulting in interference. And the output of that is shown on the right side. And it results in homogeneity throughout that slice. So as we can see on the right side, there's images of a patient's head, and you can visually see how inhomogeneous the fields are at 3T and 7T. And this is just one example of where correction would be useful, which we'll get into later.
Okay, so why exactly do we care about B1 inhomogeneity? And there's a couple of examples of like how this is important clinically. So this is one example of a patient's abdomen, and we can see that the arrows are pointing to regions where we're basically losing a lot of signal. So it's a dark region up here and another dark region here. And since it's pretty unlikely that this person is just has a hole here, in reality, we can assume that there's this artifact that that we need to take into account. So this artifact is actually caused by B1 inhomogeneity, and it's a result of signal that has been attenuated, and it results in the signal loss. So if we're imaging this person and we get this particular slice, and we're wanting to look at an organ that's in this region, this image is how it is. It's not going to be very useful. But the good thing is that there are methods to correct this. And this is an example of what the same image looks like if it's corrected. And you'll see that you can, we gain back all of that signal that seems to have been lost there. Okay, so getting more into why B1 variation leads to signal loss, we can use the example of an inversion recovery pulse. So we've talked a little bit about this in the past, but just to talk about how this pulse plays out in an ideal condition. So we'd start off with all of the signal being in M0, and then you apply a 180 flip. So all of the magnetization will become negative M0. And then based off of the T1 times of each tissue, we'll recover some of that signal. So we can try to calculate at which point we want to image to be able to null a specific tissue. So in this case, if we image at the red line, we'll basically be nulling all the signal from white matter and have signal still left from fat and CSF. But this is only under an ideal case where you apply a perfect 180. And in reality, there's some spins that have, or that see less than or greater than 180 flip. So in this case, you might have, instead of getting full negative MZ, you'll have some, a little bit less than that. And what ends up happening is that ends up shifting the point at which some of these spins will become nulled or have zero magnetization in the Z direction. And so if you image, you might not be just nulling white matter here, but also CSF. And this might not seem like a particularly big difference, but at higher strength fields, this becomes a bigger issue because this is something that becomes magnified and you see it a lot clearer. So here's an example of the same patient being imaged using one of these inversion recovery pulses. So they were scanned using both the 3T and 7T scanner. And the 3T scan looks pretty good. There's not too many big artifacts that like you can probably pinpoint. But for the 7T, there's a little bit of signal loss at the ends here. So this arrow is pointing to some of the signal loss at the edges. And this occurred because of this B1 inhomogeneity where not all the flips at the sides were able to flip the full 180. So we end up nulling some parts of this. And then in order to correct this, first you need to be able to map, well, what flips are these actually, what flips are the spins actually being flipped to? And there's a few different methods for doing this. And I'll go into like one in particular, which is phase map method. So the phase map method is similar to the double angle method, which we've talked a little bit about in class. And one of the differences is that it relies on the accumulation of phase as we're like performing the sequence. So the sequence that we'd run is a non-selective pulse, and it's 180 degrees about the x-axis, followed by a 90-degree rotation about the y-axis. So it's kind of like a 2 alpha alpha pulse. And I'll show what that would look like in this video right now. But basically, you start off with all the magnetization in the z direction, and after applying both rotations, everything will be in the x direction. So under a perfect field, I'll just restart that. So under the perfect field, everything will start off in the z direction. And you can kind of follow the my, mz graph down here. But everything will rotate from positive mz to negative mz. So we'll first play out that 180-degree pulse. And then after that, a 90-degree pulse. And what you'll see is at the end, you have zero magnetization into y or z direction, and everything's in the x direction. So this only occurs for a perfect homogeneous field if the spins actually see the 180 and 90. And if there's any type of inhomogeneity, you might get a slightly different flip. So like 175 or 95-degree flip. And so I'll show what that would kind of look like in simulation. So it starts off the same, everything's in the z direction. If you apply an imperfect 180-degree pulse, you'll be left with some magnetization in the y direction after the imperfect 180. And then after the imperfect 90, you'll be left with some magnetization in the z direction. And then using this phase that is accumulated at the end, you can kind of work your way backwards and figure out, well, what flip was that actually at? So if you do this for all of the different places, you'll be able to create these phase maps. So here's a comparison of two different phase map method for B1 and double angle B1 method. And for the most part, both of these mappings show like the relative same trend. So there's regions of a shorter or a smaller flip and a greater flip along the edges, but the place that phase map method varies is that it is able to show a larger range of angles. So you're able to pinpoint smaller changes or smaller variations in flip. And the phase map method is compatible with various sequences like GRE and EPI, and is also usable for 3D volume. So if the scan that you're wanting to perform has some of these requirements, this is a good method to use. So some cons of the phase map method is that it does have long scan times. It takes a little longer than the double angle method. And it also has a little bit low resolution. And even though 3D was like a pro, it's also kind of a con because you can't, it's non-selective, so you can't just image for a single slice. So if a single slice is one of your requirements, this is probably not the method. But there are other methods And I'll try to go over these quickly. But so we know there's a double angle method, and this one is pretty robust, but it is a little bit slower than some of the newer methods. There's also this dual refocusing echo acquisition mode, shorthand DREAM, that is really useful for multi-slice for like brain imaging. It needs relatively low, or it has relatively low SAR level, so it's useful. It's really good for human scans, and it's also very quick. So just to give an example of how quick it is, this is 16 slices acquired using the DREAM method, and all of these slices were done in 3 seconds, so very quick. So after we're able to acquire the B1 map, from there we move on to actually using it and being able to apply it to correct for the images that we have.
Yeah, so I'll be going over 2 correction methods, and this first one is image post-processing. It's one of the simplest way to do with inhomogeneity, and it does kind of use the maps that Cindy was talking about. And all it does is it applies a filter based on the map. So before I get into the maps, I'll talk about the model that it's based off of. The popular model is shown by this equation, where x denotes the measured intensity, x prime the true intensity, and alpha just denotes the inhomogeneity effect at each pixel. And figure A on the right shows a sagittal image of the head and the breast, and inhomogeneity can be clearly seen due to the bright spots and a lot of the darker spots throughout the image. And if we move forward, you could see that a B1 field map shown by figure B is what's or figure C is what's used to correct this. The simplest way is by division into equation 1 for each pixel, so that the measured intensity now equals the true intensity. And the output of that is shown on the next figure here in figure B, where you see the corrected image, and the inhomogeneity is no longer visible. So overall, this method is advantageous because it can be conducted offline, so that like time isn't an issue, and the patient doesn't have to stay in the scanner while all this is happening. But with that, it also has some downsides. It doesn't actually improve the SNR and contrast of the image, because it only uses the information that was already there. And technically, the flip angles are still not what was desired to be, since the B1 field was not homogeneous when the images were acquired. So that leads me to the next method I'd like to cover is referred to as active power modulation. And what it does is it modulates the RF transmit power for each slice position. And this paper that I'll be going over was by Claire Stewart and others who focused on the inhomogeneity along the axial or Z direction, since it was the most dominant in terms of poor homogeneity, and it's the least sample dependent. And so focusing on the methods of the paper, they started with a calibration, which was just a pickup coil, which was positioned so that accurate measurements of the B1 profile along this Z-axis could be made. And this figure right here shows the average variation in B1 measured along that axis, normalized relative to the center of the coil. So once calibrated, the B1 data could then be used for the active transmit power modulation. And for a specific pulse sequence, the values, how it worked was like for any sequence, the values of each slice position were determined, and the appropriate RF transmitter power that scaled with it were read from a lookup table, so that while the sequence was ran, a real-time slice counter could vary the attenuation of the RF transmit power. And going on to the results of this, this correction technique was tested both with phantoms and the human head at 3T. And this figure on the right shows the main signal intensity of a cylindrical phantom for a spin echo sequence. And as you can see by the dashed line, the change in image intensity is largely removed with the transmit power modulation compared to the solid black line. And if we move on to the next,
Oh, sorry.
yeah,
Can you wrap up in like 2 minutes?
yeah, almost done.
Okay, thank you.
Yeah, yeah. So and then onto the example with the sagittal plane of the human head at also at 3T. You can see the clear reduction in intensity variation over the brain. On the original image, you see signal loss on the top and the bottom of the image, but that's recovered in the corrected form. So overall, this is another method for reducing artifacts, but this time it's at the source. And it's important, especially when accurate flip angle is critical for the image that's being required. And also, moving on from that, even though this technique is used to compensate for B1 variation only along the z-axis, it's still pretty significant because the paper, it was shown that B1 inhomogeneity is most dominant along this axis. So it was a right to fix along this axis or focus on this axis. Yeah, so overall, I hope we were able to show that B1 inhomogeneity occurs, why it occurs, and due to it can lead to signal loss and poor image quality, and it can be crucial when trying to diagnose things from MR images. And as we showed it was more noticeable at higher fields, but luckily it can be mapped and corrected, and kind of the techniques you choose really just depends on the timing, the scan time you'd like, and also what you'd like to do with the images, kind of the diagnose from the images. So yeah, thanks. And then we're also happy to answer any questions.
I guess one question I had is in that method where you're sort of just dividing by the inhomogeneity map, isn't there a fear of like blowing up noise or something like that? Like if you have really small values in the inhomogeneity map, is there any sort of regularization around that?
There was another variable that included noise, and kind of that was have to be filtered out before dividing by the map. But these images, they were shown. This I guess equation was done without including the error, and they were able to show that it was still effective. So I guess their noise would have to depend on, I guess, different scans and different images, and kind of take that into play whether you would want to include it or not.
Oh, Nikki, you're muted.
Any more questions? I said. And then I might have missed it, but did you mention anything about parallel transmit? Because I haven't seen. Yeah, so really parallel transmit is the way that people are solving B1 homogeneity right now. This is kind of the way that has been used, and that's really the major way that people have been able to mitigate with field homogeneity in 3T. And then adiabatic pulses, which I don't believe I've seen here, as well as another technique that has been traditionally used to mitigate with the inversion pulses, which are completely insensitive to B1 inhomogeneity, except that they only depend on power. So you have to inject a lot of power in order to do that. But that's kind of always the trade-off. There's always a trade-off between things. So if you want to be robust at something, you have to invest something, and that's a limited resources like power. So that increases SAR.
Mickey, did you say parallel transmit was used at 3T?
Yeah, parallel transmit is used as 3T, and parallel transmit is even the scanner, like the G scanner that we have right now, it has 2 channels. It has the quadrature coil. It's a birdcage quadrature coil that you can actually phase differently in magnitude and phase the different elements, the 2 elements there, and that in order to create a more homogeneous transmit field.
So clinically, do radiologists do it?
Yeah, there's actually a way on the scanner. There's like a button, optimize. You can optimize. If you're seeing a lot of trouble, then there's a way to optimize and shift those around, and that will do B1 shimming.
Yeah, I think you're right, Mickey. There's a lot of different techniques, and I think for the inversion recovery pulse, there's like some pre-saturation pulses that can be used as well that we didn't really get into here. But yeah, I think one of our main points is like it depends on what kind of application you need and like what your requirements are for them.
Absolutely. Any more questions? Okay, well, thank you so much. Was it our last group for today?
Yeah, it's Shreya and Alfredo.
All right. Hi, everyone. Today we're going to be talking about a project that we've been working on about multi-frequency deep learning-based off-resonance correction. So as we all know, K-space, the spatial frequency domain of the image has a Fourier relationship with the image domain of the image domain, and the spatial frequency domain, or K-space, can be sampled in many different ways. And so far in class, we've mostly focused on Cartesian readouts where K-space is sampled row-wise, and Cartesian readouts have a pretty small readout time. And when the readout time is small, off-resonance effects in general are also small. However, for non-Cartesian trajectories like spiral here, readout times can be very long, and because of that, we have to consider off-resonance effects. So ideally, when we just do a Fourier transform to reconstruct an image, we're assuming that the spins resonate at the center frequency prescribed. So at 3T, ideally all spins would resonate at Larmor frequency of 127.74 megahertz. However, in reality, the resonance frequency across the sample can vary due to several different factors, including chemical shift, which is where the protons in molecules that are not water, like complex molecules such as fat, are excited. Also B0 inhomogeneities and susceptibility effects. And this causes off-resonance, and in the image domain, we can see off-resonance effects as spatial blurring. So here you can see an example of some off-resonance, and this image was acquired with a spiral trajectory, and you can see that there's blurring and also a little bit of distortion in the edge of the images here. And we can look at off-resonance in terms of the signal equation. So basically, as we've learned, this is the received signal of MRI before any demodulation occurs. So you can see the image, and it's acquired with this K space, so Kx, Ky, and it has this Fourier relationship, but it's also modulated by the center frequency, omega naught. And so if we demodulate at omega naught, we get the baseband signal. And the baseband signal is what has the Fourier relationship with the image, and that is the raw K space that we get out of the scanner. But when we have off resonance, now we get this additional term, which is this e to the minus I, and omega e would be the sort of frequency offset from omega naught at each pixel. So you have a spatially dependent omega e, and this term basically accrues phase across the trajectory as you sample in time. And this actually kind of breaks the Fourier relationship with the image. So now if you wanted to simulate this, as we'll talk about later, you can't really just do a direct fast Fourier transform or non-uniform fast Fourier transform. So the idea behind our project is basically, now what if we, instead of demodulating at omega naught, which is Larmor frequency, what if we demodulate at multiple different frequencies in a range around omega naught? So now we take this raw case-based signal from the sample, which has off resonance, and we'd demodulate at frequency offsets from omega naught. So here, like omega 1, omega 2, omega I. And then from each of these, we can reconstruct images using fast Fourier transform, or not, in this case, non-uniform FFT. And using these images, there's a different amount of blurring in each one of them, and we can harness that information to figure out what kind of frequency information in the different voxels, and hopefully use a network to reconstruct a better image. And so this actually has a parallel in normal imaging with focal depth. So basically you can see in the top, at different focal lengths, different parts of this image come into focus. So here the dog is in focus, and in the next image, the background's in focus. And ideally, you'd want an image where everything was in focus and everything was sharp. So an MRI that translates these focal depths have a parallel to different demodulation frequencies, where if this area of the image, if it's at a certain resonance frequency and you demodulate at the correct resonance frequency, you can see that area locally comes into focus. And similarly, at a different demodulation frequency, a different area would come into focus, but the other parts would be blurry. And ideally, we'd want an image that everything is in focus and everything is clear. However, MRI is a little bit more complicated, actually quite more complicated than a normal, than the step from focus problem, because first, one voxel in MRI can have multiple different resonance frequencies. For if you had a voxel with both water and fat, which is very common, you'd have multiple frequencies, but in this image, you can't have a pixel with different depths. So every pixel would just have one depth. The other thing is that in MRI, the blurring is not localized. So here in the top, you can see that when the dog is out of focus, it's not blurring on top of this couch. But in MRI, actually, when a certain area goes out of focus, it blurs on top of the other spatial locations, and that makes it a much harder problem to solve. So we'll demonstrate actually with a simple toy example. So here we've simulated two circles simulated them in case-space using a jink, and then in the image domain, that's what it looks like. But then if we assign these two circles to two different frequencies, for example, fat and water, and then we demodulate convert it back to case space and demodulate it at the water frequency and the fat frequency, you can see that when at the water frequency, the water circle is in focus, but the fat circle blurs on top of the water circle, and it's not just out of focus within its area. And similarly, in this bottom image, you can see the same thing happening. And so that's exactly why it's a more difficult problem to solve. And if we actually look at, so now instead of two frequencies, if we look at a sort of cut along this image at a range of demodulation frequencies. So this is basically if you take this line and this line, but in a continuum of demodulation frequencies, you can see that how this blurring happens along this line. So there's this sort of pattern of blurring where the blurring occurs on top of the other circle. And this is related to the point spread function of the spiral trajectory. So for example, if we had a point and we just had a single point at water frequency, and then we demodulated that point at a continuum of demodulation frequencies, you can see that this blurring actually has this pattern. In 3D, this is actually a cone pattern. And you can see because this blurring occurs not just along the line where the original point was, but it blurs along Y spatially. That is the reason why you see this blurring on top of the other parts in the image. And so in general, for off resonance effects, each voxel in the image will have a different point spread function because it can have different material. So like this, if the material was fat, for example, then this point spread function would shift a little bit. So you have this sort of spatially varying convolution because of off resonance effects. And in general, since CNNs are performing convolutions, it can be useful to do these kind of deconvolutions. So the idea was to take several different demodulation frequencies, which correspond to different lines in this image, and use it as an input to a network and train the network with that. So actually before that, conventionally, some off resonance correction methods that have been used, so some analytical methods are, the first one is conjugate phase reconstruction. And this basically, if you had knowledge of this term, so of the off resonance term in your signal equation, so that is basically the B0 field map. So if you had the B0 field map, you know what this exponential term is, and you can use that to correct for the signal equation. So basically you can apply the conjugate phase of whatever phase was accrued across the trajectory due to off resonance, and then perform some kind of non-uniform inverse DFT or some kind of segmented approximation of that to correct for the off resonance. And some limitations for that are you basically need accurate field maps, and that could be hard to get. That can be hard to acquire because field maps can be noisy when you acquire them, and they may not be exactly accurate. And also you need to perform 2 separate scans to acquire field maps. And also this method can be computationally slow because you don't use FFT. And another method that doesn't require a field map is called autofocus, where you do the same concept where you demodulate at different frequencies. But then across the different frequencies, you choose the best version. So the frequency that has the best version of each pixel using some kind of sharpness criteria, and create some kind of concatenated image with that criteria. And some limitations of that are it doesn't perform well at all with spatially varying off resonance, which we'll show later. So Alfredo.
Yeah.
Okay, yeah.
Thanks, Ray. Yeah, so basically we set up a deep learning pipeline where our input, instead of, so I guess previous work has like the demodulation at omega-0 frequency. And basically that is fed into a neural network. And the idea is like you ask the network to correct the image. But however, as Shreya was mentioning, we believe that if we incorporate multiple demodulations, maybe the network will have more information to do a better job. So the idea is we have all this input and maybe the network can learn how to choose from this, and maybe we choose some regions from here and some region from here in the bottom. However, this combination is just not just like choosing pixels, but it's also like a nonlinear combination because we have like the blurring occurs across the whole image. So the way we do it, we pick an image in magnitude, and then we go to K-space using the non-uniform FFT. And from there, we can demodulate at different frequencies. So we pick an image, we go to K-space with NUFFT, and then we can get our, I guess we use our ground truth, just going back with the adjoint. The difference here is that when we go to K-space, we are sampling with the spiral trajectory instead of Cartesian. So we can get like the spiral kind of filling of the image. So then if we want to incorporate some off resonance for training, we were training with constant off resonance. So actually we started trying to do non-uniform off resonance. It is possible to do analytically. However, it is very computationally expensive. We did optimize the function a lot. We threw it to GPU and so on, but it will just either use the whole GPU or either use a lot of time. So it wasn't feasible for training. So for training, we are doing constant off resonance, which is based on previous work and did get good results. So basically, from our spiral K-space, we can add some phase, which is some random of resonance, let's say plus 50 or 100 hertz. And then we get our K-space, which has the of resonance, and then we demodulate at multiple frequencies, for example, omega naught plus delta omega, and so on. And with that, we get a bunch of images that are going to be the input of our network. And yeah, so yeah, those are the stack of images that will go into the network. And then the network is very simple, bunch of layers I'm going to show in a bit. And the output is a single image. So yeah, the network we have, in this case, our data is complex. We separate it into channels. We have a real part and an imaginary part in both channels. And we also have n images because we have n demodulations. In our case, we were using 11. And then it's very simple. Every arrow here is a convolution of 5x5 plus ReLU. And then we have also some ResNet connections. So we have 3 blocks where each has 2 layers of 5x5 plus ReLU. And then at the end, we have a single convolution to get to our final image, which is also complex. So I guess for results, we train on data. This is a human connectome data set. So our ground truth is on the left. And the input looks something like this, where sometimes the image that is most in focus is like maybe on the left. Sometimes it's going to be on the right. It's going to be any of these guys. And the idea is that the network can kind of use one of these as a starting point and basically combine all this into a final image. So I guess in the next slide, we can see this is only the before correction at the resonant frequency, but the input has all the demodulations. We can see outputs from the autofocus, our network, and the ground truth. You can see the network is doing very well, as well as the autofocus for this case, because it's constant of resonance. We do have some kind of funky here on the background. This one was trained with the ground truth as completely dark, like the original one, without doing the spiral go to K space and back. So we actually saw better results later, but we haven't documented them. And then we said, well, why train with only brain data? Maybe we can just be a bit more general. Let's try to train with only noise. So basically, we can generate random maps of noise. And the way we do is we just generate random K-space, and then we can mask it with a with a exponential. And with that, you go back to image domain, and you get an image that will have like different frequencies. They could be like super noisy, could be less noisy. Basically kind of random, adding randomness everywhere to have like random data. We also have to kind of turn the edges dark, so we avoid aliasing. And basically this is how we train. We get a bunch of noise images, a lot of them, and then we ask the network from here, get me the ground truth, and it did a pretty good job. And the
just FYI, 2 more minutes left. Just if you can stick, yeah.
I think we can. And then the question is, okay, it does great on noise. Can it do great on brain? It did. So here we have a bunch of examples. The first one is the input on the omega naught resonance frequency, but we have all the frequencies as well. And the outputs, as you can see, we're able to do a pretty good job. Next slide, please. We also tried, well, we cannot do non-uniform maps. We can do maybe piecewise constant node maps, also doing random maps and assigning frequencies at some cuts thresholds. And basically we ask the network correct this, and it's able to do it. And I guess autofocus here is not able to do a good job. And the last thing we tried is partial volume. Basically, let's say we have 2 frequencies, for example, fire and water, and at some, I guess on the right, you can see like here, some regions are more on focus, and maybe the top images are also some on focus, more on focus here. Basically not assign a single frequency, assign 2 frequencies, and assume that some proportion of the pixel belongs to fat and some proportion belongs to water. And it's always also able to solve this. Yeah, I guess future work we want to do is better evaluation. There's something that is not quite working when we actually do in vivo data. Maybe it's our inference, maybe something else we have to look at. Previous work has done well on training on constant field map and then evaluating on non-uniform. So we have to figure out what we're doing wrong. But as we can see, in the training is doing very well. And if we can, our hypothesis is if we can bring the the original training to whatever works on non-uniform, we should be able to bring all of them as well and probably do better. So yeah, we also want to try like linear or piecewise linear. Right now it's constant, and also probably high resolution and new loss functions. And I guess finally have a question for the audience. How important is off resonance? We covered this in lecture. I don't know if Ekin can make a poll or just raise your hands for A. B, C. Okay, I see 5 hands. D. Okay, yeah, the answer is C. You're right. It is very important. I was once Mickey said it's very important. I said, oh, I want to work in this. And the thing is, like a couple of lectures later, he said something else was super important, and I got a bit disappointed. It's only very important, but I'm glad it's at least very important. So yeah, that's our presentation. I want to thank a lot Mickey and Ekin for all their help, and also Sukrit that is on here. We wouldn't be able to do this without their help. Any questions? Yep.
So yeah, I have a question. Just to be sure that I understand this, you would need to train this network for each machine, right? Because the mappings are different for each MRI machine.
Yeah, and actually for each trajectory. Here I mean, right now we're training with a specific trajectory with a specific readout length and so on, and specific resolution, everything. So yeah, we will have to train on like any different sequence you want to do. However, something we want to try later is maybe we can make a bigger network, right? That is more capable, and maybe throw random sequences inside and combine all of them, and maybe it can figure out in a more general way.
Got it. Yeah, so I was asking because how do you get the ground truth for a machine if you don't know the B0 mapping? Like what do you use for the ground truth?
Yeah, so right now we start with a clean image that was not acquired actually with Spiral. That was acquired with a short readout like Cartesian. And on there we simulated the off resonance, and that's how basically got our training first.
But also like for in a machine, you can do a spiral with a short readout, and then that would have very minimal off resonance. And that's sort of what we had been doing for our in vivo data, and then doing a normal spiral. So a shorter spiral, but many interleaves, and then you can acquire a fully sampled but image with very minimal off resonance.
Yeah, I see. Thank you.
I mean, actually, I have a question following up, Daniel. Is off resonance depend mostly on the machine?
Depends. I mean, we have
not really.
No, well, it depends here a lot of the. I mean, yeah,
There's a spec for a machine. The machine is spec'd.
I mean, the machine has some level, yeah, some ppm, but we also have a lot of
1 ppm is the machine. But what creates off resonance?
The body.
Right.
So like water, we get inside, we modify the whole field, right? And if it's what butter or what, whatever fat, it's also going to be different. So yeah, that's a biggest source.
More questions?
I have a question, and it's like more like a general question, or both for this one and the motion correction one. So there are a lot of work on using retrospectively simulated corruptions like motion or off resonance. But when you really apply it to like real scan data, it sometimes fails. What do you think is the most important thing to build this gap?
I guess for us, we think right now it's realistic simulations. So since right now we're training on these piecewise constant off resonance, but real field maps will be smoothly spatially varying and maybe not fit this model very well. So maybe that would be some way to have this translatability.
Yeah, definitely the data, the distribution of the training and testing has to be either the same or the training cover the testing. Like noise is much more broader and ideally covers the testing that will be the brains and actually what we do in the scanner. Yeah, like if training right now we're doing constant and then maybe it doesn't generalize for non-uniform, maybe that could be an issue. So yeah, definitely make sure you don't have subtle crimes in your training, going JPEG and stuff. And yeah, basically distribution should match.
Okay. Well, thank you, everyone. We're a little bit, I think, over, but that's okay. Thursday, we have how many presentations?
5. So
We
one
have
more
5
than
presentations.
today.
So please be on time, not 10 minutes after. Let's start on time so we end up like now. Okay.
Okay.
All right. Well, thank you, everybody, all the presenters and everybody. And yeah, see you on Thursday, everyone. Thanks again.
Quick question. Should we send like the wiki page that we edited or?
Yeah, absolutely. That's
do-
part of your submission. Yeah.
Oh, put it through. Great. Okay. Thanks.
Yeah,
Thank
I'll
you.
make like a thing on Grayscope that you can put it down.